Hepatitis C virus using 454 pyrosequencing, and it's a collaboration between my lab, TIMC MAC in Grenoble, and the University Hospital in Grenoble. So, okay, so a very basic introduction. Viruses, self-replicating infectious agents. Technically, they are dead outside a living organism, but once they infect a host, they can use the host's resources and multiply. So they are made of RNA, composed of RNA, and one of the main features of these viruses is that the genome is very variable. There are lots of mutations in a very short amount of time, and the viral population within the host can change rapidly. So this is a huge challenge for drugs which target specific areas of the viral genome. So since it has so many mutations, most of the drugs they fail to act on the infection. So my project, we're using next generation sequencing and statistical methods to basically identify, measure, and study the viral diversity, and then try to see what the impact or what this diversity has, the impact of this diversity on the antiviral therapy. So moving on a bit about the virus, it's quite widespread worldwide, and you have traditional bitherapy of two antiviral agents, and there's a new therapy which adds a protease inhibitor to this bitherapy. So what it does is it acts on a specific region of the genome, which is the NS3 region, and it actually it prevents the replication of the virus. And that's how it tries to control the infection. But the problem with this therapy is that it fails. So if you see this graph, on the y-axis you have viral load in a patient, and there are timed samples. So each sample is taken after 4 weeks. So if you see, this is a non-responder of the treatment. So the patient was treated for using the tritherapy and bitherapy, but for both of them it failed. So in the beginning the viral load was this much, and at the end of the treatment, when the treatment was stopped, the viral load returned to the same level as the beginning. That's why it's a non-responder. So as you can see, both the blue one is the bitherapy, pink one is the tritherapy, and both of them follow a similar curve, that they reduce the viral load in the beginning, but as soon as the treatment is stopped, it just goes back to normal. So the question is whether this is related to the diversity of the viral population in the host. So how do we measure this viral diversity, and how will we study this? So for that, we need to extract the HCV virus from the patient. So there are two sources for these samples: the plasma of the host and from the blood, and the cells. So the plasma are PLV, CCs are from the cells. So this is a simple phylogenetic tree of a mixed sample of PLVs and CCs. And one unique thing you can see here, that the blue ones, which are these from the cells, they form a completely different part of the tree. They are quite distinct from the sequences which are extracted from the plasma. Now this is quite weird, because it's the same virus which has infected in the same host, but still two different sources of the virus, they have a different, quite a different distinct sequences. So which one, can we get some information from this? Something like, are the CCs the one responsible for the antiviral resistance, or are the PLVs, and are there specific clusters within the viral population which actually cause the treatment to fail? So that's why we study trees, but the problem with trees is it's too big. So this one has around 900 sequences, and it's quite difficult to actually pinpoint clusters and try to get any meaningful information. So I'll get back to this challenge of using clusters later on. And so coming on to the aims, first of all, identify the known mutations in samples which actually cause the antiviral resistance. Then measure the viral diversity and try to see if it is related with the outcome of the treatment, whether they are responders, non-responders, and see that. And then try and understand the structure of the viral population to see if there are clusters which cause the antiviral resistance. And yeah, and there's one more thing. As I showed here, the CCs and PLVs, they are quite distinct. But if you see, there are few blue lines in between. So that is a question from the clinicians, and it's a biological question that the two sources, they are different, yet they have some common, mixture of sequences. So we are trying to find out whether there's a real distinction between these two or whether there's any mixing. So coming on to the methods, the sequencing part, it was performed in the hospital, and I just used the raw data of these, but I'll just explain a bit about it. It's amplicon sequencing. The whole gene is sequenced at a time. That is the main target of the antiprotease therapy. So we have 40 patients, and each one of them has different samples, like in the beginning of the treatment, after 4 weeks, after 8 weeks. And for the CC and PLV comparison, there are 20 patients. So moving on to the entropy. Entropy is basically the measure of diversity. It is a single number which tells you which represents the diversity in the sequences. So how do we calculate that? That's calculated using the frequencies of mutations obtained from the GS Mapper software, and then using a Python script to change the formats. And finally, this formula, it's used for calculation of the entropy, and that is done using a Perl script. And this is done for each sample, each different time sample. So coming on to this method, as I had shown you earlier, the big tree, and it was quite difficult to see the clusters. So this is the same tree, and as you can see, you can't really make out any clusters within the tree. And because each tree will be different, and your definition of a cluster might not be the same. So for that, we use this statistical method called PCA, Principal Component Analysis. And it essentially represents this phylogeny in a 2D graph, and the same graph is represented here, and you can count the number of clusters. There are 8 phylogenetic groups, and they are all differently colored, so it's easier to visualize the clustering within the tree. So this, and then finally we have the results. The first result is we found out the entropy of the non-responders was much higher than the responders. So the main conclusion we get from this is that yes, higher the viral diversity, that is higher the entropy, more there's a chance of the patient being a non-responder. So then coming over to the PCR results. So PCR gives us 2 kinds of results. First is to check whether the CCs and PLVs, whether they are similar or distinct. So if you see this graph, the blue ones are CCs, yellow or PLV, and they are quite different. There's mostly there's good distinction between them, but there's some area here which is a bit ambiguous. So we are not sure whether it's either of the groups. It might be a mixture, and this is what we saw in the first graph. There are a few blue lines in the tree which were not grouped with all of the other blue lines. So, and we have some phylogenetic clusters again which are seen to be mixed. So this is essentially the same graph. These are the CCs, and here you can see the CCs. But if you see here, I'm not sure if you can see the blue dots, they are composed from both of these, both from these and these. So there is some mixture between the two groups. Then coming over to the conclusions, yes, viral diversity is correlated with the outcome of the treatment. Then there is compartmentalization in the viruses. CCs and PLVs are quite distinct. And finally, clusters, which we saw, they might be either exclusive CCs or PLV, or there might be some mixing. So moving on from this, what we can actually know from this. So first of all, try to pinpoint the source of the antiviral mutations to see whether any single cluster is responsible for the failure of the treatment. So essentially trying to remove the bad guys. And then another practical result is that the clinicians, they find it quite difficult to extract the CC sequences. So they want to know if it is worth actually trying to extract them. So if we don't find any antiviral resistance mutations in the CCs, they won't perform the analysis, and it will significantly speed up the treatment and diagnosis. So, and finally, yeah, if some clusters exclusively confer the antiviral resistance. And okay, that's all. Thank you for your attention.